All right, thank you everyone for coming to the presentation of Bridge Biotherapeutics. My name is Pavel Princev and I'm the Director of Business Development. As a public company, the usual disclaimer applies and my statements today may contain some forward-looking statements. Today I'm going to quickly briefly introduce our company as well as a high-level overview of our pipeline. So our company was founded in 2015 in South Korea. We also have a US-based, independently run research center in Boston, known as the Boston Discovery Center, or simply the BDC for short. The South Korean headquarters is responsible for the R&D of our clinical pipeline, as well as setting the strategy and governance for the organization as a whole, while the Boston Discovery Center has their own early-stage drug discovery pipeline focused on oncology assets, as well as a drug discovery platform. At the beginning of when we were founded, our company was a very small team of industry experts, about a handful of people, and together they in-licensed 3 promising preclinical stage assets that they were able to develop through IND approval and bring them into the clinic where they are today. Since then, we've doubled in size to about almost 50 people, and we are now approaching 4 clinical stage assets. We hope to have 7 by the end of 2025. And then from 2026 onwards, we see ourselves to be on the path to becoming a more fully integrated global biotech company with an established sales network in the United States. So here's a quick snapshot of our pipeline that I'm going to walk through today, kind of grouped by our indications of cancer, fibrosis, UC, and then at the end of the presentation, I'll introduce a company that we recently acquired, which is an ECL biosensor diagnostics company. So starting off with our oncology assets, a very area for a lot of high unmet need is in the area of non-small cell lung cancer, particularly for patients who have progressed on 3rd generation EGFR TKIs, who possess a C797S mutation. We have 2 such 4th generation EGFR TKIs in development. A quick overview of this indication here. So for lung cancer, it continues to be the most common cancer and the leading cause of cancer death in the world, with over 1.5 million cases diagnosed every single year. And among those cases, about 85% or so are of the subset non-small cell lung cancer, NSCLC. And if we look a little bit further into the genetic drivers of non-small cell lung cancer, we see that EGFR mutations make up the majority of these genetic drivers. In Asians, it's responsible for about 50% of cases, and for Caucasians, up to 15% of cases. Looking a little bit further at the types of EGFR mutations responsible for this, we see that about 90% of these mutations or so are in the form of DEL19 and L858R mutations, and these driver mutations are typically treated with EGFR TKIs. And depending on the generation of EGFR TKI that the patients receive, they will inevitably progress on these EGFR TKIs, and they will have either a T790M mutation, a C797S mutation, or both. And C797S mutations are exclusive to progression on 3rd generation EGFR TKIs, for which there is no currently approved viable treatment option available. Osimertinib, as you are well aware of, has already been on the global market for about 7 years or so. And then in other regions, there are other 3rd generation EGFR TKIs that are in their respective local markets as well. And each year, the amount of patients progressing on these drugs due to the C797S mutation is steadily increasing and accumulating. In more of the developed nations, frontline use of osimertinib is becoming more prevalent due to the evolving NCCN guidelines. However, this is not always the case for other countries. In countries particularly where the prevalence of EGFR mutations is particularly high, osimertinib is still given in the 2nd line. And for those areas, we see a high prevalence of patients with the C797S containing triple mutations. This makes the need for a 4th generation EGFR TKI with sensitivity against C797S and T790M very important, and both of our 4th gen EGFR TKI assets are able to meet that global need. Very recently, we concluded our BID dose escalation cohort for the BBT176. In the QD cohort, we had 2 partial responders. One of the patients was a patient presenting with a DTC triple mutation. They had a 51% reduction in tumor size, Another patient was one with an atypical del19 driver mutation, and they had a 30% reduction in tumor size. BBT-207 is a structural derivative of BBT-176, and its broad spectrum activity against the most common EGFR mutations allows it to be positioned in a more earlier line setting, ideally for patients who have progressed on osimertinib from the first line, and some of the preclinical data here suggests that there's activity in the front line setting as well. So for BBT-207, just a couple months ago, we presented an in-depth preclinical update at the AACR conference I don't remember where that was, but anyways, I was there. Yeah, it's too many of these conferences. So for those of you who are interested to learning more about that, I highly encourage you to find our poster, and much more information will be there. Other updates, just a couple months ago, we got our US FDA clearance letter to start our first in human study for this asset, and we plan to start the global phase 1 trials very soon in a couple months. Moving on to the IPF projects. So IPF, or interstitial pulmonary fibrosis, belongs to a large group of about 200 or so lung diseases known as interstitial lung diseases, and they're commonly associated with scarring and fibrosis of the lung. And as the name idiopathic suggests, there is no known singular cause of this disease. So being able to explore multiple independent pathways that are associated with the progression of this disease allows us to combat IPF from multiple directions. So BBT-877 is our most advanced IPF asset in our pipeline. It is a first-in-class autotaxin inhibitor. Autotaxin is an enzyme that is found in higher concentrations in patients who have certain fibrotic diseases and inflammation, as well as fibrosis of the lung. And autotaxin, what it does is it catalyzes the hydrolysis of LPC and speeds up the local production of LPA. This LPA then binds with several LPA receptors, triggering a downstream signaling cascade, leading to epithelial apoptosis, which is one of the primary pathologic events in moderate pulmonary fibrosis and IPF. And BBT-877's selectivity for autotaxin inhibits the production of this LPA, greatly slowing down the progression of this disease. And the charts here on the right are from our phase 1 clinical trial, where patients were given BBT-877 in a multiple ascending dose, and we were able to see that patients who took BBT-877 at 200 milligrams twice a day were able to have a reduction in LPA levels of about 90% when compared to baseline and steady state levels. And this, compared to some of the leading competitors for autotaxin inhibitors, BBT-877 is one of the most efficacious. There's been a lot of developments with BBT-877 over the past year. We had the FDA approval for this compound in July, and just last month we dosed our first patient in our multinational phase 2 study, and we expect to have the clinical study report sometime toward the end of next year for this asset. 301 and 209 are two of the preclinical stage assets that we in-licensed last year. They're both in IND-enabling studies at the moment. Like most of our pipeline, these assets are first in class. BBT-301 is a potent potassium channel modulator, and BBT-209 is a selective GPCR19 agonist. Similar to 207, we have presented a more detailed preclinical data for these compounds at the IPF Summit here in Boston last year. So once again, please find that poster to learn more. And finally, I'll end the presentation with the company that we acquired, Ellipse Diagnostics, which is a next-generation ECL biosensor diagnostics company. This company was founded in 2021 by Ik-Soo Shin, which is he's a professor at Sunshine University in South Korea. And a lot of the developments that they have been doing in their lab led to the discovery of this company, which has been able to break down a lot of the limitations of ECL biosensor technology that is really limiting the true potential of this technology. So to really kind of understand and appreciate what it is that they have done, a quick run-through of what this biosensor is and how it works. ECL is essentially electrochemiluminescence, and the biosensor, what it's doing is it's able to detect and quantify how much of your target of interest is in a particular sample. And it does so by attaching a very selective bioreceptor to your target of interest, and that bioreceptor is tagged with something that's going to emit light. So we just call it a luminophore, as it's indicated in that legend over there. And in order to get that bioreceptor to emit light, it has to react with something, right? And what it's reacting with is simply a co-reactant. And currently, of all the ECL biosensor devices that are available in academia and industry, there's only one co-reactant being used at the moment, and that's called TPRA. The issue with TPRA, this is the bottleneck in this entire system. The bioreceptor is extremely sensitive, your detection device is extremely sensitive, but the co-reactant is the problem. And the issue with TPRA, it has poor solubility in the actual reaction medium, which limits at the very, very small sensitive scales for target detection. Also, because of its chemical and physical properties, it's very difficult to handle and store. It decomposes under certain conditions. So that all of that translates to limitations in sensitivity. So what the group was able to do here, they were able to eliminate all of those negative things associated with TPRA with a new co-reactant. Here it's indicated on the chart on the right in orange as CR, because this manuscript hasn't been published yet. But when they simply replaced TPRA with this new co-reactant, they were instantly able to increase the target detection limit over 200-fold. And this brought the limit from picomolar to picogram per milliliter to femtogram per milliliter, which is hasn't been done before. And this has made this particular biosensor one of the most sensitive on the market today. And our goal with this is, through the acquisition of this company, to further strengthen our own development capabilities, as well as to partner with academia and industry who utilizes these own analytical methods in their own labs to potentially add a new revenue stream to our company. And finally, closing remarks. Again, thank you everyone for coming. And if you have any questions, I'll be outside, and then I'll be happy to take them there. Thank you.